The maize crop in the Kuching division have recently been subject to a persistent and unidentified insect pest, leading to considerable concern among local farmers. In response to these reports, our team from the Department of Agriculture conducted field investigations to collect larvae from the affected maize plant as illustrated in figures 1 and 2. Despite through food assessment, the specific pest could not be identified on site. To resolve this issue, we have decided to implement molecular identification technique, focusing on the COI gene as a barcoding markers. This method is expected to provide a precise identification of the pest species, which is essential for devising effective pest management strategies. The following section details the DNA isolation protocol using the CTAP method, which will be employed to extract high-quality DNA from the collected larvae. Following DNA isolation, polymerase chain reaction (PCR) will be conducted using COI-specific primers to amplify the DNA, which will then be analyzed via gel electrophoresis. This molecular approach will enable us to accurately identify the pest and support the development of targeted intervention to protect the maize crop in Kuching division.
Nanti bikin 2 minit.
Terima kasih.
2 minit. Tuh, singlet. Okay. Kai.
Electrophoresis is performed by placing enzyme digest DNA samples into wells generated in a gel and placed in an electrophoretic tanks equipped with positive and negative electrolyte, which stated by EMIT 2021. The purpose of gel electrophoresis in DNA marker analysis is to separate and visualize the DNA fragment of different size. DNA extraction and amplification would be the first step. DNA would be extracted from the collected larva in specific regions, such as the mitochondrial COI gene commonly used for insect identification, would be amplified using the PCR. The amplified DNA fragments would be subjected to gel electrophoresis. This process separates DNA fragment based on size, with smaller fragment moving faster through the gel. The separated fragment forms distinct band that can be visualized using DNA stream. By running this sample alongside known DNA markers or samples from identified insect species, researchers can compare the band patterns. The unique fingerprint created by the pest DNA can be analyzed and compared to database of non-insect pests. DNA barcoding, when combined with sequencing, allowing for precise species identification, even when morphological identification is challenging or impossible, as in the case of larva or lineage specimens. Once the pest is accurately identified, appropriate pest management strategy can be developed and implemented, potentially saving the maize crop and elevating the farmer. There are 3 observations, which is the first one is band present, where the visible bands in the lens indicate the presence of DNA. The second is band intensity, which the bands are indistinct, indicating that the DNA concentration may be low. And the lastly is unclear band, where the unclear bands can be a result of a variety of reasons, which including low DNA concentration and inadequate staining or insufficient UV exposure times, where it may also indicate DNA deterioration. So next is PCR protocol. The polymerase generation, which is known as PCR, is an in vitro method in which DNA sequence or transcript are quickly amplified with very high specificity and fidelity, utilizing the oligonucleotide polymerase and text DNA polymerase in a simple automate, which stated as Hadidi et al. 2017. The result did not show any DNA fragment on gel agarose. Since COI polymerase is not being used to trim the DNA, it is deposited at the end of gel as shown in the figure.
So next, in this video, I will talk about the difficulty on the experiment. In the experiment, we encounter and experience some difficulty during the preparation of the experiment. So the first one, a proper grinding of the sample. The sample need to be grind properly until we get the desired sample form. This is to make sure the accuracy of the result. The liquid nitrogen added slowly to make sure it is in a solid condition. Next, the second difficulty that we get and experience during the experiment is that a safety in using the chemical. The experiment should be handled safely and carefully to avoid and prevent any accident, especially involving the chemical. The third one is during the gel preparation. The gel should be fully dissolved during microwave phase. Then it should be cooled down a bit before pour into the tray. The bubbles should be avoided to get a perfect gel. During the experiment, the challenging part was the removing comb part. It must be removed carefully to prevent a gel from broken. The fourth challenges or difficulty that we experienced during the experiment is that during the pipetting and phase separation. Pipetting is the crucial part in this experiment. The pipetting should be handled carefully. Avoid handshaking and stay focused during the pipetting was the difficult part during the experiment. Hand need to be steady and stay still to get a good result. The fifth part that we encountered during the experiment was the bubble. During the extraction of the sample or protein from the tube while pipetting, the prevention of bubble is the most difficult part. The pipetting process must be done slowly and precisely to get a good result. The last one is that to maintain a low temperature of the sample. During the sample extraction, during pipetting, the sample must be kept in a low temperature. It is to make sure we put back the sample in the ice after pipetting the sample every time we use it.